Good afternoon, everyone. So, welcome. This is an outstanding show of plankton appreciation today. I'm so impressed. I'm not surprised, but I'm very impressed. I can't believe how many people have said, "What a beautiful day for a lecture." So, I'd like to begin by acknowledging that we at the Marine Science Center, oh, by the way, I'm Diane Quinn. I'm the director of the Port Townsend Marine Science Center. I'd like to begin by acknowledging that we live and work on the traditional lands of many Coast Salish people, including and particularly the Sklallam people, the Strong people. We respect and are grateful for this land, these waters, and the people who have inhabited them since time immemorial, and who continue to steward this place for future generations. I'd also like to say that this series, Future of Oceans, is now in its 10th year, and that is in no small part thanks to the Darrow family for their, thank you so much, where are you? Thank you guys. For supporting this series and lifelong learning in many, many ways. And as Susie Darrow told me, they're partial to plankton. I also want to mention that Gretchen was on the Nature Now series on KPTZ, interviewed by Nan Evans. Is Nan here? No, not right now. So we'll put the link to that interview on our website when we post the video from today's talk. So if you want to go back and hear things that she talked about with Nan that maybe she isn't talking about today, that would be great. And we also just want to thank Nan Evans for supporting the series all year. She's interviewed our speakers on Nature Now, and you can hear all of those interviews from our website. Okay, so today's speakers are both faculty at Washington State University. There are still people coming in. And that might be Nan Evans. I just said her name, and look, there she is. Yay! I love when that happens. Thank you, Nan Evans. Both in the, I'm going to use my big notes here, both in the School of the Environment at Washington State University. They came up from Vancouver, Washington today. They are co-directors of the, make sure I say this right, Aquatic Ecology Lab at WSU Vancouver. But their paths intertwine quite a bit, but they are separate people with separate research and other career goals. And so just a little background if you didn't read the bios online. Gretchen Rollwagen-Bollens is now an associate professor at WSU, and she spent 8 years at Woods Hole. 4 of those years, at Woods Hole, but working for the Sea Education Association, and 4 of those years were spent on a 135-foot schooner teaching oceanography to undergraduate students for 9 months of the year. And if you kind of follow through her resume and her publications and what she's doing now, you can see that this support and love for teaching undergraduates is still part of what she's involved in and really champions. She is the director of the Southwest Washington Regional Alliance for Inclusive Science Education. That is a collaboration and partnership between WSU Vancouver, Clark College, and Lower Columbia College, which really focuses on breaking down barriers for students who may or may not find their way and be supported through science careers in college. And her other research line is really looking at how supporting undergraduates and even high school students in discovery research is an indicator of if they will stay in the sciences, if they will pursue careers in science and stay in the sciences. And that's a really important thing for our current scientists to be doing. And so besides her other research and field work and everything else she's doing, that's a priority for her. So in addition to plankton work, her particular field of research right now is looking at harmful algal blooms, and so people in the audience who are working for doing sound toxins research for us and for everyone, this will probably really resonate with you. And Steve Bollens, I didn't even say, so she went to school at Harvard for her undergrad, University of Hawaii for her master's, and, I was gonna remember this without, UC Berkeley for her PhD. So it's pretty impressive. Steve Bollens started in Florida, did his undergraduate work at Oberlin, and then his master's and PhD at University of Washington, but his research and teaching appointments have been pretty broadly spread. He was also at Woods Hole for part of his career, but also at University of Washington, San Francisco State, and then engagements in New Zealand, Australia, and Germany. So now to have landed at WSU Vancouver, he is also the director of the Myers Point Environmental Field Station. So between the two of them, they are teaching thousands of students and doing their own research and really deeply committed to finding ways to connect the science of what they're doing to practical areas that the rest of us can understand a little bit more easily sometimes. Like, how we're looking at shoreline armoring or other environmental issues. What does oceanography have to do with topics like restoration ecology or conservation biology? And so that applied practice is also part of what they're doing. And today they're going to really focus on plankton, and which, as I told a whole group of people last week, this is the most important thing in the world. So good thing, you're all here. So plankton, Mysterious World of Plankton is going to be way less mysterious after we get to hear from both of them today. So please welcome our speakers today, Gretchen and Steve. Gretchen, Rollwagen-Bollens, and Steve Bollens. Thank you very much.
Thanks everybody for being here, especially on such a gorgeous day. This is going to be, in case you're wondering who is who, I'm Steve, this is Gretchen. And this is going to be a presentation in two parts. Gretchen is going to start with the broader, more general introduction to the mysterious world of the plankton. And then I'll take over and dive more deeply into a narrower but still quite fascinating issue around cascading migrations in a fertile fjord.
Let me also thank you for being here and that very gracious introduction. I'm tired just thinking about it. I'm going to advance. So I'm going to be giving, I think most of the people in this room, based on what I'm hearing, probably know as much or more than I do about plankton, but I wanted to give just a broad kind of little march through some of the most interesting taxa and some of the most interesting things they do in aquatic systems. And I'm going to be focusing primarily on marine systems, but I work almost entirely in freshwater nowadays, so I'm not going to give them the short shrift. So first, I'm just going to throw up a few images here that you may think, what do these all have in common? And some of them, you probably recognize most of these. Probably you're not going to be surprised to know that connects all of these organisms and even these ecosystems is plankton. And that can be directly or indirectly, and indeed the fact that we're all sitting in here breathing oxygen is another example of how we as humans rely on the work of plankton. So I want to just talk briefly about that Diane mentioned. I work for Sea Education Association, and I did the Sea Semester program when I was in college, which is a completely transformative experience for me, introduced me to oceanography. Up to that point, I was kind of a Cousteau kid, right? I'm dating myself, but was really interested in marine mammals and dolphins, and I wanted to go to sea. And when I had that experience going out to sea for months and months at a time, and actually studying and conducting oceanographic research with students, I became introduced to these incredible organisms that I really didn't know anything about before that. And so I quickly went from being I'll just call it a dolphin dreamer. I'm just looking for alliterations here. And now I consider myself a PPP, a person passionate about plankton. And I have a feeling that I have some compatriots here. And I'll just note this is a pteropod, a marine mollusk, pelagic mollusk, and it's even smaller than that next to these little dolphins. So I want to just take a little bit of time to tell you a little bit about some of the major groups of planktonic organisms, and I'm going to do this in the context of how they fit into a food web. So if we start down at the lower part of the slide, I'm putting two sort of broad groups of phytoplankton. So these are planktonic organisms that float freely in water and are capable of photosynthesis. And so these are the primary producers, and I've just grouped them into the large ones and the small ones, right? And with green. These guys are really important sources of energy and materials to zooplankton. Now, zooplankton, even though the word zoo suggests animal, zoo in this broad sense actually means any organism that has to consume something else in order to get energy or absorb organic material directly from the water itself. So it's animals, it's protists, it's bacteria, it's a whole group of consumer planktonic organisms. So I'm grouping them into the small ones. These are mostly single-celled organisms. And then there's some medium-sized ones, and you might recognize a copepod there. They graze upon these phytoplankton. They can also graze upon each other. And then copepods I'm just kind of keeping at the center because they are a really important component of the diet for a lot of larger zooplankton, again, consumers, but at higher trophic levels that are carnivorous sometimes, they're omnivorous. They have a whole bunch of different feeding strategies, but there's these large organisms. And then they are typically consumed by fish at varying stages of their life history, some larger invertebrates, but most of the energy is moving from those phytoplankton at the base and moving up into fish and then whales and other organisms that I'm not putting on this slide because they aren't as important. I'll also just mention that there are a lot of larger zooplankton, and when you get above about the size of a copepod, which is about 1 to 2 millimeters, then everything's kind of size structured below that. Big things eat little things. But when you get to a copepod and above, big things can eat little things can eat big things. And the relationships are really complicated and fascinating. So I'm putting a lot of gelatinous plankton here, which also can be consumed ultimately by these top predators. So I'm just going to start at the bottom and just give you a few, some little vignettes about these guys. So talking about the small phytoplankton. So we've probably all heard of cyanobacteria before, or blue-green algae. These can come in single cells that are shown here, or they can be in these colonial forms, either in filaments or in these long chains. One of the things that is really unique about cyanobacteria is that this is probably the most ancient organism on Earth. These are stromatolites. They look like rocks, but they're actually communities of cyanobacteria that gather and accrete different sediments and things together, and they become very, very hard, but they grow. The outermost part of this is layer over layer over layer of living cells. And these are the first organisms. They date back about 3.5 billion years. So they were the first ones to really contribute photosynthesis and converting CO2 into oxygen and helping to oxygenate our atmosphere. The other thing that's really pretty cool about cyanobacteria is that they have the capacity to, we call it fixing nitrogen. So they can access atmospheric nitrogen, which is a molecule that's very, very hard to break down. It's two nitrogens that are really bonded tightly together. And so unlike most of the other phytoplankton, all phytoplankton need nitrogen and phosphorus and silica in order to in order to grow and photosynthesize, like fertilizers. If there isn't enough nitrogen in the water, then the cyanobacteria can look to the atmosphere, and they can start accessing atmospheric nitrogen, converting it into a biologically available form, and then outcompeting their other phytoplankton peers. They also are really important in moving carbon around from the atmosphere into the water and then burial in the deep ocean. Cyanobacteria in freshwater systems and some estuarine brackish systems are also the source of a lot of different harmful algal blooms that we can talk about later. Another group of really interesting but small phytoplankton are the coccolithophores. And this is a satellite image. This is an actual photograph. It's not computer generated or false color. And this is the coast of somewhere in the North Sea. And this white milky stuff, those are actually millions and millions and millions of these little cells that are made of calcium carbonate. And they have these little plates that they build around the cell. The actual organism is somewhere tucked inside. And so this is an electron micrograph photo that shows those plates. Each one of them is made of calcium carbonate. When the organism dies, it sinks and takes that carbon with it down into the deep ocean, which is, again, one other component of taking carbon from the atmosphere through photosynthesis, incorporating it into bodies, and then those bodies sinking into deeper depths. And that's just to illustrate that atmospheric carbon through CO2 is taken up in the process of photosynthesis, but there's also a chemical reaction going on. And indeed, as our oceans become more acidified, then that's contributing to this complicated sort of dynamic between you take up more CO2, the ocean absorbs more CO2, it becomes more acidic, and then it becomes very corrosive for that calcium carbonate. So it can be a real problem that these coccolithophores are really important for taking up that CO2, but they're being corroded a little bit more by that increasing acidity. In the larger phytoplankton, so we're still in photosynthetic organisms, diatoms, these are really common in all aquatic systems, fresh and marine. They have a frustule or little skeleton that one component is silica. And so in order to grow, they need nitrogen and phosphorus like everybody, all the other photosynthetic organisms, but they also need silica, and they form these beautiful, really highly complicated and porous skeletal systems that allow them to exchange materials from inside the cell to the water. But there are these really beautiful patterns. And around this area, you may be familiar with domoic acid, which is produced by Pseudonychia, which is a diatom genera that when there's times of the year when you see those signs going up and you shouldn't go out and collect razor clams, or they're affecting marine life. And that's due to high numbers and big blooms of these Pseudonychia that produce this toxin. Another group of phytoplankton, again, these are all just cool, so I'm just going to keep listing them for you. Dinoflagellates, they come in a couple of different forms. Some are armored. They have these cellulose plates, but these are still single cell. These are organisms that are just composed of one cell. They can be unarmored, but they have this very characteristic shape, or at least there's a sort of a waist, a groove going around the middle, and then there's one that usually goes up and down, and they have flagella in there that in a moment I'll describe what they do. They can be highly motile, but one other thing that's kind of neat about photosynthetic dinoflagellates is that they can bioluminesce. So they actually produce these chemicals that when the chemicals are brought together, they actually react and produce a little bit of cold light, so it's blue in color. So this image here, this is what the cell looks like, and this is what it looks like under a light microscope, and this is what it looks like when it's been stimulated to release those chemicals. And so that leads to these big bioluminescent blooms that sometimes you can see very visibly. They also, though, there are a lot of dinoflagellates that produce toxins, and so they also are a group that we're concerned about, thinking about harmful algal blooms sometimes that can suck up a lot of oxygen and can lead to these fish kills. Okay, well, let's talk about the things that eat phytoplankton. These are my favorite groups, actually. So I'm going to start with the small ones. These are all still single-celled organisms, but they're really complicated. So these ciliates, what defines them, probably not surprising, is a bunch of cilia they have right around, usually sort of an oral groove or an oral opening that allows them to swallow prey directly. They're transparent usually, so you can see what's going on inside them. They can be sort of in this form here without any kind of outer covering, but they also can form what looks like a shell, but it's actually a lorica. It's made out of materials that are sort of organic materials, and then they can also, they can be, what happened there? Be sticky and pick up little bits of sediment, and then they live inside them. And they use those cilia to create water currents to pull suspended other plankton for them to consume. The other thing about ciliates is they're kleptomaniacs. They're actually, they perform a process called kleptochloroplasty, which I ask you to say 10 times quickly. What they do is, so you see this in panel A here. There's a ciliate that's all red on the inside because it's been consuming a little photosynthetic cell, much smaller than it, a little flagellate that has red pigment in it. So it's not that the organism is red, it's swallowed a bunch of things that are red. But it's gotten some nutrition from that, but it's retained the chloroplast, so it still can do photosynthesis. So these guys are kind of having their cake and eating it too. They can consume other prey as consumers, and they can get energy from that, but they also can continue to use the products of photosynthesis from those little algae that they ate and kind of stole their chloroplast. But in this figure here, you'll see that it's red to begin with, then it starts consuming these green little flagellates, and after a while it loses those red, it sort of scoots them out, and now it's become greener because now it's full of green diet. So it's a really interesting strategy to be able to survive under conditions when maybe there's not a lot of food to eat. Fine, I'll just eat something that can make its own food, and it'll be good. And something I didn't know until I prepared this talk, actually, is that ciliates can do the same thing with macroalgae. Not just little floating flagellates, that there are these, this is a plant called, I'm not going to remember it right now, but anyway, this is a standing macroalgal, seaweed. That macroalgae, which is shown here in panel B, produces these little swarmer cells. It's part of their reproductive process. And so this ciliate here, which is what those black dots are, they're sort of surrounding that macroalgal component, the thallus there. Those ciliates are eating those little swarmers that macroalgae produces. And that little red spot there is red pigment. And then you can see inside this ciliate that red pigment is retained as well. So that kleptochloroplasty can also happen with macroalgae. And so I'm talking about the small zooplankton. No, there's dinoflagellates. I already talked about them. This dinoflagellates actually sometimes can be photosynthetic, sometimes they can be consumers, and sometimes they can do both. I'm going to talk about the ones that are consuming other things. So here you can see that morphology a little bit better, where they've got these armored plates and these grooves, and they have these little flagella that move inside them. The coiled ones make them go like this, and the long ones make them go like this. So they swim in these helical motions. They're highly modal. They can consume a lot of different things, usually in this process where they essentially stick out a little straw called a peduncle. And so they can actually consume things that are as big as they are or even bigger because they use extracellular digestion, where they essentially stick out a straw. This is the dinoflagellate here. It's grabbed this spherical organism. It's stuck the straw inside the organism. It's another little ciliate, actually, and it's just sucking out all the interior parts, and now it's inside the dinoflagellate. Rotifers are a freshwater taxa. They do the same thing. They eat each other a lot, so there's a lot of really kind of scary sort of interactions happening here. This large rotifer here will consume smaller rotifers. Now we're getting into multicellular organisms. And these rotifers can be found in all kinds of places wherever water will collect. So you can find them in pitcher plants, you can find them in old tires. Copepods, you're going to hear more about copepods in the second half of our presentation here. These guys are crustaceans, so they're arthropods. They have an external exoskeleton, they have jointed appendages. Copepods, some of them have a really complicated life cycle. They start as larvae here, and there's multiple molts. They actually get rid of that exoskeleton, they grow a little larger, then they exude another skeletal layer. Whoops. And so they go through about anywhere from 12 to 15 stages in their life cycle. They also have this really interesting way of feeding. We call them suspension feeders because they're living in this three-dimensional medium and their food is floating all around them. Their particles they want to consume are suspended in the water column. So at the size that we're talking about, they can't just reach out and grab what they want to eat because it's so viscous that every time, think if you're in a pool of honey or something, every time you want to reach for something, you're just going to push it further away. So they have the capacity of using a lot of their appendages to make water currents that actually then pull water with the suspended food in it towards their mouth. And in this case, it's a wonderful confocal, this is a confocal image of a real copepod. Its mouth part's about right here. They've got these little spines or setae on their mandibles, and when they rotate those around, they pull water towards them and into their mouth, and then they have a bunch of swimming legs as well. So they're really very, very active, and that's a common way for crustaceans, actually. I want to just make brief mention that there are a lot of larval stages of a bunch of different invertebrates that are planktonic in their larval stage, but then they metamorphose and they grow into larger adults, and they usually sink out of the water column, so they live in the benthic zone, or in the case of freshwater, oftentimes they can be insects that then fly away. But they come from all kinds of different groups, so this is just a blast of larval images to give you a sense of the diversity. I do study freshwater now too, don't hate me. So Daphnia are really common genera of Cladoceran. There are some marine forms of Cladocera, but they aren't quite as prominent in marine systems as they are in freshwater. And one of the really interesting things about daphnids is that in response to the environment, they can actually change the shape of their exoskeleton. These are also crustaceans. And they do that in response to the presence of predators. So it's we call it an inducible defense. So you can see that the little spike on the top of this guy's head gets longer and longer, and so does its tail. Makes it much harder to eat. And we can see other, development of spines and things like that. So if they're in the presence of, if they somehow sense that there are predators, they will, over their own lifespan, make changes to avoid being eaten. Euphausids, krill, these are very common in marine systems. They can form these big bait balls, sometimes they're called. So they're all swarming near the surface. So every red color there is a euphausid. And then they could be taken advantage of by, this is a fabulous photograph of a blue whale that then is coming up and taking advantage of that swarming behavior and being able to swallow thousands and thousands of these krill or euphausid organisms all at once. So they're really important transfer of energy from the plankton into our largest organisms. Amphipods, I just had to include these for Phoenibas because they're also really interesting and they're kind of creepy looking. These are amphipods, also crustaceans. Everything looks kind of like a shrimp. They actually they will capture, sometimes it's a living organism, sometimes it's just the little house it's left behind, but this is a little salp. And it's taking over the salp body, which is this gelatinous organism, and then it's now living inside that. And so it usually has its babies there and eventually then uses that little house and protects it for its lifespan. Pteropods, these are the ones that really got me to be a person who's passionate about plankton. They're mollusks, but they're entirely pelagic. They have a foot that actually acts more like wings. They're called sea butterflies. And they also, they're beautiful. If you see them live under a scope, they're really just fascinating. They will eat each other, and they have these ways of capturing others of their, not necessarily usually the same species or genera, but other pteropod forms. Chaetognaths, it's also going to be something that's going to come up in a moment. These are arrowworms is their common name. They may be an inch or two long, so you can see them without a microscope generally. But if you had a microscope, this is what you would see, that their head, they have these spines that are also a sensory organ, and it allows them, they have them on their head, and they also have them along the upper part of their body. They are voracious predators. They will eat anything that they can grab onto and then start chewing. And so that will, I'm just giving you a little hint that might come up again. And then finally, all these gelatinous organisms that kind of defy, well, I'm going to take that off for a second, defy this size structuring. But they can be kind of small, they can be kind of big, but they'll eat things that are much larger than themselves. They don't have mouths to be able to consume a whole organism. They'll stick to it and they'll use chemicals to break them down. All right, so I am now going to pass it on to Steve, who's going to be talking about cascading migrations.
So thanks, Gretchen. So that's, again, a broad general overview of the diversity of form and function in plankton. And I'm going to be doing a sort of deeper dive on a particular aspect of the life history of plankton, that is cascading migrations in a fertile fjord. And we have the good fortune today to have the author of this book in the audience with us. Richard Strickland wrote this book in 1983, I believe it was. An excellent introduction to the plankton of Puget Sound. And so I recommend this to you. It's written more for the lay person, but really, really fascinating work. And I shamelessly borrowed his title to use in the title of my talk, although the fertile fjord in this case is not all of Puget Sound, as in Richard's book, but more specifically Dabob Bay, Washington, which I'll tell you about shortly. That story really starts with Bruce Frost, a professor of oceanography at University of Washington. He was not the first one to undertake oceanographic study of Dabob Bay, but he was the first, I believe, to do it in a very systematic and long-term way. So he had a small army of graduate students, of which I was one, who looked at the ecology of Dabob Bay and all its various aspects, including and in particular plankton ecology and zooplankton even more specifically. So I wanted to acknowledge and thank Bruce for his efforts. So to orient you a little bit, we're here in Port Townsend. Seattle is off to the southeast. Dabob Bay is pretty much just due south of us here, an extension of northern Hood Canal. And to zoom in on this a little bit, we've got Quilcene and the town of Dabob to the north, Coyle and Brennan to the south, and Dabob Bay is shown here. And there are two parts of this bay in particular that I'm going to be speaking about today. One is a so-called deep station. And when I say deep, I mean quite deep. 190 meters, that's over 600 feet. This is as deep as the Space Needle is tall. And I'll contrast that with a second station up north that I'll refer to as the shallow station. Shallow is a relative term here. It's 60 meters deep, 200 feet, that's a 20-story tall building. So I'll be using the terms deep and shallow, but again, these are relative terms here. Both are quite deep. The main player in this are copepods, and Gretchen has told you a little bit about them. Here's one species in particular that's very abundant in Dabob Bay, Calanus pacificus, and I want to draw your attention to these large antennae here. And what is kind of hard to make out in this lighting is that there are these tiny little hair-like structures along the first antennae, setae, that are mechanoreceptors. They can sense movement in the water. Similarly, these antennae have little holes or pores in them that allow the copepod to sense different chemicals in the water, so chemoreceptors on their antennae. And then thirdly, these copepods are cyclops. They have a single eye in the middle of their head that is not an image-forming eye, such as you and I have, but it can sense light and dark, so relative light intensity shadows, if you will. So between being able to sense mechanical disturbances in the water, chemicals, some light variation, they can sense their environment in many ways. So now I want to talk about a particular process or phenomenon called vertical migration. And this is the process by which many, not all, but many zooplankton reside deep in the water column here during the day when the sun is out, and they only come up into the surface layer to feed during the cover of darkness. Now, there's two important things about this migration pattern I want you to know. One is that it's utterly massive. This is often referred to as the largest, most ubiquitous migration on our planet. This occurs in the oceans, it occurs in lakes, it occurs in rivers, it occurs in ponds, the world over. So many, many millions of metric tons move on a daily basis, typically down in deep water during the daylight and then up at night. It's absolutely massive. The second thing is that it's very impressive. These copepods, an average length of 2 millimeters, I happen to be about 2 meters tall, so I'm 1,000 times larger than these copepods. These copepods can move 100 meters every day, every evening and every sunrise. They move up and then back down. That's comparable to an organism such as you and I moving 60 miles, 100 kilometers every sunset and every sunrise. So these organisms are small, they're moving huge distances, and just the amount of biomass that is moving vertically across our planet is huge. So, very interesting. At least I think so. But why? Why are they doing this? What is the potential advantage to these migrators? And the two common theories have been to avoid predators. If your predators are visual and they need the light to see you, better to hide in the deep, dark waters during the day and only come up to feed at night. Another set of theories around saving energy. These are so-called cold-blooded or poikilothermic animals. Their respiration is directly proportional to the temperature. So if they move down to cooler waters, their respiration rate declines. There's an energetic bonus, or so the theory went back in the day. So ubiquitous phenomenon, diel vertical migration every 24 hours up and down. But why? What's the advantage to this? And so we started with a series of field observations in Dabob Bay, including this first one from a 2-year study in spring, late April of '85. And this shows the vertical distribution of Calamus pacificus during the day, these open distributions during the day, and during night. And what you see here is most of the copepods are in the upper 50 meters, upper 75 meters, both day and night. There's really not a lot of migration going on here. There is some small proportion of the population that's moving up, and so I calculated an index here of the proportion of the population. About 5% is moving up from depth during the day to the surface at night. A very, very weak migration, or indeed no migration at all. So that was 1 month, April. A couple of months later, stronger migration. These copepods are deeper during the day, and about a third of them, 0.33, are coming up at night. A couple of months later, stronger migration. They're deeper during the day, and most of them are coming up at night, about 3/4, 74%. Then A couple months later in October, a slightly weaker migration, but still down during the day, up at night. Next year, similar but different in some respects, same late spring date, but half the population is migrating vertically. A couple months later, still about half. A couple of months later, much stronger migration. 79% of the population is moving up and down every night. And then by October, almost all of the population is migrating. So this migration is variable. It depends on the season, it depends on the year. And that led us to think, why? What is driving this variation in the migration? So we undertook more field studies, and in particular, we wanted to know what the fish were doing in Dabob Bay. So we took a series of midwater trawls and surface tow nets, and we sampled the fish in Dabob Bay at both the deep and shallow stations. And amongst the most abundant fish there were Pacific herring, very common, very abundant in the surface waters in Dabob Bay. Likewise, juvenile salmon, this is a coho, there were 4 different species, a fairly large number of juvenile Pacific salmon in Dabob Bay. And next, this may surprise some of you that are familiar with this fish, Pacific sand lance as an adult burrows into sand in very, very shallow waters. But as juveniles, it's entirely pelagic, meaning it lives up in the water column in the surface waters. So what do these 3 most abundant fish species in Dabob Bay have in common? 2 things come to mind. First, they live in the surface waters. There are lots of fish that live on the bottom. These are pelagic fish. They live up in the surface. They're visual predators, and they eat, can anyone guess? Plankton in general, copepods in particular. So these are so-called planktivorous fish, very, very abundant in Dabob Bay. So when we looked at the variation of this migration on the left, sometimes weak, sometimes medium, sometimes strong, and we compared that with the abundance of fish in Dabob Bay, we found this correlation, this statistically significant relationship between strength of migration, weak here, strong here, and the abundance of fish, low to high. And there's a relationship between when these copepods migrate and whether or not there are fish present. The strength of copepod diel vertical migration is significantly correlated with the abundance of visual predators. Pretty clear, right? So are we done? Is that it? Not entirely. So this next point may be obvious to some of you, but I think it's nonetheless quite profound, and that is correlation is not causation. So just because 2 things are correlated, in the case of the figure I showed you just a moment ago, the strength of vertical migration and the abundance of fish, it doesn't mean that one is causing the other. Lots of things are correlated, some of them for causal reasons, others for random or so-called spurious reasons. And the gold, how would you then test for a causal relationship? How do you go from these field observations where you're looking at correlations between different environmental variables to an actual test of the causal relationship between one and the other? Well, in science, the gold standard for this is a manipulative field experiment. A manipulative experiment. Could be in the field or could be in the laboratory. But what you do is you hold all things constant and you just manipulate one variable, let's say fish abundance, and you look at the response variable, in this case, copepod migration. So that's what we did. It took us a long time and a lot of effort, but we not only undertook the correlative field studies, we went and did the manipulative experiments as well. And one place we did this was at the Max Planck Institute for Limnology in Plön, Germany. They have 10-meter tall plankton towers around which they constructed an entire building and experimental infrastructure. And I had the good fortune of spending a couple of visits there using these towers to test experimentally the ideas that we had generated from the field studies. Similarly, just outside of Friday Harbor Laboratories, I undertook these enclosure studies where I use an enclosure, a large plastic bag, And we manipulated fish, had some fish in some enclosures, no fish in other enclosures, and we were able to look at the effects of migration. And finally, at our laboratory at WSU, we have so-called plankton mini towers that are shown here with the video microscope that spans the vertical extent of each tank and can map out the distribution of the copepods and their food, et cetera. To summarize many years of hard work, not just Gretchen and I, but our graduate students, et cetera, I want to show you this series of cartoons here. So first is a case where there's lots of fish in the surface, but no phytoplankton at all. And what we see here is that the copepods are down during the day and up at night. Strong vertical migration with fish, no phytoplankton, no food. Second cartoon here. When we remove the fish completely, but we put lots of food in the surface, the copepods immediately, not over time, but they immediately move up to the surface day and night. These tiny little organisms, so-called primitive organisms about the size of a grain of rice or sand, have this complex infrastructure, mechanoreception, chemoreception. They can sense their environment, both predator and food, and they respond accordingly. Third cartoon, when we have both fish and phytoplankton presence, but we manipulate the relative magnitude such lots of fish, low phytoplankton, they immediately become migratory again. The copepods do, down during the day and up at night. And then if we reduce the fish but add some phytoplankton, there again, the copepods are non-migratory in the surface. Again, the behavioral repertoire of these so-called primitive animals is incredibly sophisticated and complex. Okay, so I want to then just move on and quickly give you two case studies of so-called cascading vertical migrations. And this first case study is from Dabob Bay, where a slightly different copepod, a smaller copepod called Pseudocalnus, and it's invertebrate predators in Dabob Bay. And here's an observation of the vertical migration of this copepod at the shallow station now, so down to 60 meters. The copepods are all deep during the day and then up at night. This is so-called normal migration that I've shown you several examples of. Well, we contrasted that, and I want to acknowledge Mark Oman, one of Bruce Frost's other PhD students. He observed, the group observed, that at the deep station, this same copepod, just a couple of miles away now, was up at the surface both day and during the day, and then actually migrated down at night, the so-called reverse migration. So normal, down during the day, up at night, reverse, up during the day, down at night. Same bay, just two different stations that vary in depth. What is going on here? Well, at the deep station, there are invertebrate predators, and lots of them. Gretchen talked about them. Keating's gnats with these horrific spines that are very effective at capturing prey. Euphausids or krill that also eat copepods. And these invertebrate predators have the following vertical distribution in the deep station. They're down during the day, up at night, down during the day, up at night. In other words, these invertebrate predators have the so-called normal migration, and their prey, the copepods, have the reverse migration. So a predator is doing one thing, and it cascades to the prey, which does the opposite pattern to avoid getting eaten. So this is one example of cascading vertical migrations. And the other concerns this copepod, yet a different copepod, Acarcia hudsonica, and its modal prey, Akashiwo here, a dinoflagellate. And in the case of the copepod, here's a series of night, day, night, day, night distributions. The copepod is shown here in the black triangles, up at night, down during the day, up at night, down during the day, up at night. What does its prey do, this modal dinoflagellate? It does exactly the reverse. Down at night, up during the day, down at night, up during the day, down at night. So again, a second example where the predator, in this case Acarcia, undertakes one type of migration that cascades to effects on the prey, which does the opposite migration. Okay, so interesting, fascinating, I hope you agree, but who cares, or why should you care? Is this just some sort of behavioral curiosity, ubiquitous as it is across our globe, our planet? Why should we care? And the answer, in a nutshell, is this very complicated figure that I'm not going to belabor, but the so-called biological pump. And that is how the biology of the ocean removes or pumps carbon dioxide from our atmosphere. And some of you have probably heard this is increasing in our atmosphere. And biology draws it into the ocean, transforms it, and leads to eventual burial in the sediments. And of all the various processes that are important to the biological pump, I'll just highlight vertical migration here. But it's a process I've just been talking about for the last 15 or 20 minutes. And because zooplankton come into the surface, they feed on phytoplankton, and then they migrate to deeper water where they respire, they defecate, they're consumed by predators. This vertical migration is a process that actively transports carbon from the surface down near the bottom where it can eventually be buried. So in the face of climate change and the global carbon cycle, we should be very interested in the role of vertical migration. It's estimated to account for about somewhere between 10 and 30% of the vertical flux of carbon in the ocean. So not only is it interesting and curious, it's also very important. So quick summary here. As Gretchen described, tremendously diverse plankton are, both in form and function. They have this migration pattern called diel vertical migration that's clearly related to predator avoidance, but it varies depending on the type of organism, its habitat, the time of year, et cetera. And there's this even more complex behavior around cascading migrations, where the migration behavior of a predator, such as a carnivore, can affect, does affect the migration behavior of its prey. And finally, these complex and fascinating behaviors are quite important to the flow of material and energy in the global carbon cycle, and so should be of interest to us for that reason as well. Last slide, just want to go back to Dabob Bay and make 2 quick points. I think of Dabob Bay as a living laboratory for coastal oceanography. It was selected as such by Bruce Frost and his colleagues 40 years ago. It has continued to be a focus of scientific study. Lots of people are aware of the cultural significance of the bay, the economic significance, the aesthetics of the bay. Not everyone is aware of the scientific significance. And I just a couple of days ago did a search on Google Scholar. How many publications worldwide are there that make a reference to Dabob Bay? 2,610. So it has had an outsized influence on how science generally, coastal oceanography, has been done over the recent decades. So I encourage you all to conserve and preserve this natural treasure. Last slide, for those of you that are gluttons for punishment. If you want to hear more about copepods and copepodologists, Charlie Miller from Oregon State University just published this book last year, Oar Feet and Opal Teeth. And it is a deep, deep dive into copepods. Thanks for your attention and interest, and Gretchen and I are available to answer any questions that you have.